All right, so welcome everybody to our Freshwater Aquatic Invasive Species Workshop. Thank you for joining us today. My name is Vivian Matuk. I'm the Environmental Boating Program Manager for both the California State Parks and the California Coastal Commission. And I'm going to be leading this workshop in partnership with our colleagues from different agencies, including California State Lands Commission, and also with the Division of Boating and Waterways and the California Department of Fish and Wildlife, and also the California Fish and Wildlife Services. So thank you everybody for joining us today. Couple of meeting rules. I'm going to keep everybody muted. The reason why is sometimes these systems tend to create a little bit of background noise, and it's not that we don't want to hear your questions. You're going to have opportunity for that purpose, but it's just to keep the workshop really enjoyable for everybody. We're going to ask you to please ask questions only during the devoted time for that purpose. And also to please refrain from using the chat. I can check the chat, but sometimes that creates a lot of distraction, especially for the speakers. That's why we have created an agenda that will help you to ask questions whenever you need to do that. In addition to that, let's exercise. We are all adults, of course, respectful listening, and everybody is encouraged to participate, and please be present. Sometimes the fact that we are joining virtually tends to allow us to multitask, which is, not okay. So hopefully you take advantage of this great opportunity of having experts on the topic and ask those questions. Some workshop etiquette. I want to encourage everybody who can to have your cameras on. That will give us more like personal touch to the workshop. Sometimes your signal is not good or unstable, or you don't have a camera built in your laptop. So we totally understand that. And again, in order for you to unmute yourself, feel free to click on the microphone, or if you're joining us by phone, which I don't think that's the case for a lot of you, please dial *6 to unmute, and the same for muting yourself. Again, just only ask questions during the section about questions that we have devoted right after each presentation. You can unmute yourself, raise your hand, or also use the chat during the question and answer section of each presentation. So during the registration, I ask you your county of residence and also your background. And I wanted to share with all of you today this map, which shows a very good representation of different counties all over the state. So we're very excited to have this amazing representation. And we even have a couple of people joining us from Florida. So it's great to have even that national impact via these workshops. And as you can see on the listing to the right, we have state and regional parks. We have state agencies, of course, colleagues with the marinas, with the yacht clubs. In addition to that, we have Coast Guard Auxiliary, Power Squadrons, water utility and conservation districts, harbor associations, a couple of nonprofits, marine businesses, muzzle dogs, recreational boaters of California, dog owners, general public boaters, anglers. So a fantastic group of water enthusiasts and people interested in this topic. As we clearly stated in the information that you received that brought your attention for this flyer, or for this workshop, sorry, via the flyer, today we're talking about freshwater invasive species. We're going to start from a bird's eye view perspective with our colleague Cesar Blanco with the United States Fish and Wildlife Service, talking about why we need to care and what are some of the impacts that these invasive species have in our state in terms of cost, operational impacts, and also environmental impacts. Then we will have the section devoted to questions, and then we're going to start talking about key invasive species that are impacting our state and what the state is doing. So we're transitioning to our colleague Lydia Kenninson and also Michael Rodriguez. They are going to be talking about the Division of Boating and Waterways Aquatic Invasive Plant Control Program. And also, we're going to start transitioning a little bit into the quagga and zebra mussel world, talking about the prevention grant. Then we have questions, a break, and then we're going to fully get immersed into quagga, zebra, and the new invader for the state, unfortunately, which is the golden mussel. We're going to be talking about vectors, how they got here, and what the state is doing. But most importantly, we want you to leave the workshop with some action items that you can implement at your local level to help us to stop the spread of these invasive species. After that, we're going to have the section for questions. We're going to wrap up the workshop and we're going to ask you for your feedback via our evaluation form. So that's it for the introduction and the overview of the agenda. Again, check the chat where I included the links to the two handouts, including not only the agenda, but also the online resources that we're going to be sharing with you today. So let me stop here sharing the screen, and now let me introduce to our colleague, Cesar Blanco with the U.S. Fish and Wildlife Service. Again, we're going to start from a bird's eye view perspective about regional invasive species issues, cost, operational impacts, and potential problems. So welcome, Cesar.
Good morning, everybody, and thanks for being here. Happy to see everybody's smiling faces on screen. Before I get started with the presentation, I wanted to ask folks something about invasive species costs. And so this is a poll that you guys can vote on. And the question for your consideration is, cumulatively, brown tree snake invasions have cost the world how much? Is it 100,000, 10 million, 10 billion, or 10 trillion?
Thank you, Cesar. So everybody should be seeing right now in your screen the poll. If you're not very familiar, feel free just to read it and answer it directly. We're going to give a full minute. We have 35 seconds here right now, and we want to see full participation. We have a good group today, so make sure that you please are participating in the poll. And we will stop the poll and share it with Cesar so he can give us all, as part of this educational workshop, the latest and greatest in terms of this poll. All right, so let me end the poll here and share the results. It's all yours, Cesar.
Yeah, so we have a pretty informed audience here. Obviously, A and D were pretty far out there, and so there was a competition between 10 million and 10 billion. But the actual correct answer, and the key is cumulatively, brown tree snake invasion has cost the world $10 billion. So, this drives home the point that in the invasive species world, prevention is the best strategy to begin with. Make sure they don't get here in the first place. We don't have to deal with them. So I'm going to go ahead and share my screen. Okay, can you see that, Lydia? I'm sorry, Vivian?
Not yet. And for people who have bad connections, we all have good connections, so probably it's on your end.
How about now?
Perfect.
Okay, hold on 1 second here. Trying to get well, I'll just go ahead and start. As Vivian had mentioned, my name is Cesar Blanco. I'm with the U.S. Fish and Wildlife Service. What I do is I manage the Aquatic Invasive Species Program in the Pacific Southwest region for the Fish and Wildlife Service. That region covers California, Nevada, and the Klamath Basin. And the first thing I want to do is to just go over a summary of what I'm going to talk about. I'm going to describe, what are invasive species. I'm going to talk about costs associated with invasive species, the pathways of establishment and spread. I'm going to give you some key examples of invasive species and the problems they cause. I'm going to talk about some specific invaders of concern. I'm going to discuss some tools and then also what you can do as a citizen to ensure that you aren't facilitating the spread of invasive species through the Golden State. So here's the actual definition of invasive species. It's an organism that causes ecological economic harm in a new environment where it's not native. Some traits that all invasive species have in common is that they're fast growers. They're very aggressive in their competition with the native flora and fauna. They reproduce rapidly. They have a high propensity to disperse. They are morphologically and ecologically very adaptable or plasticity. They are very generalist in their requirements. And most of the time, since they're foreign, they don't have a native predator that will key in and prey on them. So, they go on largely unchecked. In the San Francisco Bay and the Bay Delta area, this area is one of the most heavily invaded regions in North America and hosts more than 210 introduced aquatic invasive species. We have endured over 150 years of exposure to large-scale introductions of non-native species through a whole host of means: commercial shipping, aquaculture, bait industry, sport fishing, water diversions, and recreation. And in the last decade, a new species has arrived about every 12 weeks. Luckily, many of these do not take hold, but those that take hold obviously pose major problems for our environment. With regards to costs, they're, every year the introduction of non-native invasive species has been increasing. Collectively, these introduced species have tremendous impacts to many things we value, and globally, the cost of invasive species amounts to about $423 billion annually. Invasive species play a key role in 60% of recorded plant and animal extinctions. And as I said before, brown tree snake, together, actually, brown tree snake and bullfrog alone have cost $16 billion globally. In the U.S., the reported invasive species costs reached $1.28 trillion from the period of 1970 to 2017, with an annual average cost of about $26.8 billion. So those are some of the costs. Let's move forward to how they get here. Aquarium imports, aquaculture, ballast water, intentional introduction, live bait, live seafood, aquascaping. Right here is a picture of a Australian red claw crayfish that was introduced in several parts of Nevada for essentially black market aquaculture. You can see why someone may want to eat one of these guys here. But so, for example, golden mussel, the pathway was determined to be ballast water. More on that later. We've seen all kinds of carp species in the eastern United States invading some of the major rivers out there. And there have been examples of gizzard shad in the Colorado River Basin being introduced through aquaculture. And this was actually lawful aquaculture, not black market aquaculture. So how do they spread once they get here? Aquarium releases, stowaways, boats and equipment, shipping crates, recreational activities, infrastructure maintenance, and field activities. Here's a picture of a large goldfish found in Lake Tahoe, I believe. There's also been an example recently in the San Joaquin main stem of an aquarium release. It's a large-scale loach. It's a common aquarium pet that someone released into the San Joaquin River, and it's taken hold and is continuing to spread at a very slow rate. And it hasn't posed any problems yet, but that remains to be seen. Hopefully they can eradicate it before it causes any more problems. But all kinds of recreational activities, you know, if you've been in Lake Tahoe, they have a very strict boating decontamination and inspection program there. And oftentimes, some of the folks, professional biologists in the past, have been guilty of spreading invasive species as well. So here's a specific example of how zebra mussels were potentially, luckily none of these zebra mussels were actually viable specimens, but there was a potential here for there to be zebra mussels spread throughout the United States vis-a-vis these what are called moss balls. These are sort of pet thing that folks like to have on their at their office desks and stuff like that. You know, they're very visually pleasing and all that, but it turned out that these moss balls were contaminated with zebra mussels here, as you can see there. These tainted specimens were found in PetSmart, Petco, and they were found online on an app called Wish. And they were found in over 26 states, including Alaska. They came from Ukraine, from a distributor in Ukraine, and nationwide efforts to stop the spread were successful. And in fact, recently there was another instance where additional shipments were captured at one of our major ports. And once again, there were zebra mussels in these specimens as well. So it continues to be a problem, and we need to stay vigilant. And several agencies, USDA and my agency, the Fish and Wildlife Service, coordinated to stop the importation and are using eDNA to find contaminated specimens as a tool. Okay, so moving on, here's some brief examples of some invasive aquatic plants in California. Many invasive plants outcompete young native plants for space and nutrients. They have deep tap roots and more efficiently retain water. Our native plants have difficult time adapting, and as well as our native fauna to the alterations that these plants pose. When invasive plants take over, the results can be devastating for many native plants and wildlife that depend on them. Common traits that these invaders possess is they can form dense mats here. Here's an example of alligator weed forming dense mat there that can actually cause anoxic conditions, meaning zero oxygen underneath this dense mat, and kill off all kinds of fish. The invasive plants can reproduce asexually. They can grow extremely fast. They can tolerate a wide range of water conditions, and they're very aggressive. I'm sure if you've seen a lot of stream roadsides, many people have seen giant reed, arundo. Some of the problems that they cause is they can clog entire lakes, waterways, and infrastructure. They can interfere with water navigation. They can displace and reduce the diversity of native plants. Since they form these large monocultures, they support a lower abundance and diversity of invertebrates, and they're less valuable food source for waterfowl. I mean, waterfowl are not adapted to these plants, so they don't see them as food source. As I mentioned before, they deplete dissolved oxygen levels. They can increase the amount of sedimentation that occurs by slowing down the flow in our rivers and streams. And then that also can cause problems with regards to flooding. Some non-native pathogens are the chytrid fungus. It's a fungus that basically attacks the skin of amphibians and causes problems for them. White nose syndrome in bats, that's another fungus. Whirling disease, which is a parasite in fish, and viral hemorrhagic septicemia. So some of the characteristics of these pathogens is they're catastrophic to afflicted populations. You know, the native organisms aren't adapted and are therefore very susceptible and vulnerable to these pathogenic infections. Many of them tend to be specific for species or genera. They can have the ability to persist in the environment and spread from location to location very easily. I mean, here's a picture right here of a invasive disease causing catastrophic die-offs in fishes. Here's some examples of invasive faunal species: the Asian clam, Chinese mitten crab, New Zealand mud snail, Florida water snake, or what we call Nerodia, zebra quagga mussels, and the latest invader, golden mussel. More on that later. They too have some of the same traits. They're aggressive competitors. They have a high reproductive output. They can form extremely high density. They can alter the ecosystem dynamics, and they're generalists and can tolerate extreme conditions. Just a case in point with the Asian clam is that it is been connected to the pelagic organism decline in the delta, because basically it filters out the water so well that many of the native organisms out in the delta, which are pelagic feeders, meaning they feed throughout the water column, the Asian clams have filtered out so much that now there's very little pelagic food in the water column, and a lot of these native organisms are dying off due to starvation. So problems that non-native invasive species cause is obviously economic, damage to infrastructure, and a loss of resources. They're the second leading cause of native species extinction and decline. I think I mentioned like over 60% of endangered species can be tied to effects of invasive species. Tend to have a loss of global biodiversity. which is not good. And then there are some human health risks. Two examples of Asian lung flu and West Nile virus. Here's another example of how a invasive species can cause a cascade of deleterious effects through the food chain. In 1996, there was a number of mysterious bald eagle deaths, and the cause of death was avian vacuolar myelinopathy. And this AVM was caused by a toxic bromine compound formed in cyanobacteria, which were tied to an invasive plant, hydrilla. So essentially what was happening is the hydrilla were providing a substrate for the cyanobacteria to grow on. Those cyanobacteria were then producing bromine, and coots were eating the bromine heavy hydrilla that was being bioaccumulated in the tissue of the coots, and then the eagles would attack the coots. Presumably, these coots were, near death and seemed like a good food source, and that bioaccumulated in the eagles to the point of being lethal. And so that's just one example of how an invasive species could disrupt a native food chain. So here's some notable California invaders of concern. Obviously, the state has put, actually, the state and federal governments have put a lot of money into trying to stop the spread of zebra and quagga mussel. Same thing for New Zealand mud snail, although New Zealand mud snail was recently found in Lake Tahoe. Nerodia or water snake is actually a native in the eastern U.S., but it's highly invasive and outcompetes native snakes out here in the western U.S. I already mentioned Asian clam, mitten crab. In the early 2000s, several areas were overrun by mitten crab, but it seems like these crabs have just sort of died off mostly. And then, of course, our newest invader that there's a whole talk dedicated to later on in this workshop is golden mussel. So here's an example of a invasive weed, alligator weed. It's native to South America. It was introduced via ballast water. It can form dense mats and can impede navigation and recreation. It spreads via fragmentation. I mean, a small fragment right here can lead to a large, dense mat like that. And it also causes problems because it can create a vector for mosquitoes. And then that's bad for our human health. Here's another example, Caulerpa, the saltwater seaweed that grows very fast. It's native to waters of the Pacific Ocean, Indian Ocean, and Caribbean. Since it's a known invader, it's been outlawed in terms of the aquarium trade in California. In 2000, an aquarium release led to a Caulerpa taxifolia invasion in Huntington Beach, and that cost nearly $7 million to suppress. It took 6 years to eradicate. And more recently, Caulerpa prolifera has been found in Newport Bay in 2001 and in San Diego Bay in 2003. And both those efforts to eradicate Caulerpa are underway. Asian clam, I think I already mostly went into this, but it was introduced in the 1800s for human consumption. And as I mentioned before, it was pointed to as a possible cause of pelagic organism decline in the delta. Chinese mitten crab, I think I mentioned that. Don't really need to go into much detail other than to point out that this is a picture of the one of the, I think, a state or federal collection facilities near Tracy, where it was overrun by these mitten crabs during their heyday of invasion. New Zealand mud snail. This invader can form extremely dense populations of 750,000 individuals per square meter. They're tiny, and they're very good at outcompeting natives for space and food. They can tie up the nutrients in the fish will feed on them, but basically they have this opercular flap that they can close and become indigestible to fish. So fish will eat them, won't get any nutritional value, and can cause them to be, cause for them to starve. Since they form such dense populations, the shells can block pipes, filters, and grates, and there's biocontrols that are underway in terms of trying to figure out how we can prevent them from spreading and eradicating where we find them. So here's right here a picture of someone's wading boot, and you can see that these zebra mussels are extremely tiny and can be overlooked. The rodeo water snake, I guess what I have to say about this is that efforts are currently underway by the state of California to get rid of water snake in California. So here are some of the tools that professionals can use to detect invasive species. There's a process called EDRR. It's essentially early detection and rapid response. And basically that philosophy is like, the first line of defense is to prevent. And then the second line of defense is to detect the population early. The sooner you detect it, the higher the probability of eradication success. And so that's why there's that rapid response component. Once you detect them, it's incumbent upon us to try to respond as quickly as possible to stop the spread. There's a newish tool out there, environmental DNA. Essentially, the old style DNA was, you had to purify it to look for your target species. This takes the opposite approach, where you collect a water sample and you basically resolve every bit of DNA that's in that sample, and it'll tell you the suite of species that are in that sample, including perhaps the invader that you're looking for. And so in instances where there's no actual visual evidence or specimens collected, you could potentially detect an invader before it gets to the level of being detectable by sheer numbers. So the next tool that folks use is particularly for those of us in natural resource management, is hazard analysis and critical control point planning. This is essentially a process whereby you look at all your activities that you partake in the field under the lens of, do any of these activities increase the risk of spreading invasive species? There are all kinds of wildlife inspectors and, canine dogs. I think muscle dogs were mentioned out there. Actually, my supervisor was out this weekend out there at Rancho Seco, and he ran into a person out there with the muscle dogs group. But dogs can be tremendous detectors of invasive species. Of course, another tool is vehicle and boat inspection stations. There are all kinds of global databases out there available, and I think that there is a sheet that Vivian has provided to you folks, or will provide to you, regarding a lot of the resource materials that you can reference to educate yourselves more on invasive species. Another tool is chemical treatments. And then, of course, outreach, coordination, and collaboration are essential because not one agency can do it by ourselves. So it takes a team. What you can do as a private citizen is just to be mindful and be aware that invasive species are out there and plan any sort of recreation accordingly, and develop good behaviors with regards to invasive species. So you don't become a inadvertent vector for spreading invasive species. There is a hotline that if you report, if you suspect there's an invasive species, you can call this hotline, and then take an active role in the spread and spread the word about invasive species. Obviously, folks here on the call today are very interested, but you spread it to folks who are just out there in nature, kind of blundering through things and not really being mindful. It's incumbent upon all of us to help spread the word about invasive species. So that's all I have, Vivian. I'll stop there and pass it on to, I guess, a Q&A session.
Yeah, thank you so much, Cesar. Great bird eye view perspective from these invasive species. So you guys learn about some of the ones that we're going to be covering today, and also some of the environmental and economic impacts we are facing due to those invaders. And I want just to open the opportunity to all of you to ask any questions to Cesar about this section we just started with. So feel free to either raise your hand or include the question in the chat, or unmute yourself, and we can go from there. To then move into, as you saw in the agenda, more specific examples of some of the species we're going to be covering today. Any questions for Cesar? Just taking time here just to check. No questions? Everybody doing okay? All right. Well, thank you, Cesar. It seems that everything has been clear, so now we can just start getting into specific questions. So we're going to move into the Delta region. I know a lot of you, based on your registration and your background, of course, are very interested in the different aquatic, the floating and submerged invasive plants that are impacting the Delta. So I want just to welcome both of my colleagues, Lydia Kenninson and Michael Rodriguez, with the Division of Boating and Waterways. So Lydia, the group is all yours. Again, I'm going just to let you take it over from there, but do you want me just to start first with the poll?
Sure.
All right. Excellent. So stop sharing here and let me now go to Lydia. So we have another poll for you. So I just launched it. Lydia, can you see it?
Yes, I could see it.
Okay, perfect. So again, the goal of these educational workshops is just to kind of do like a pre-check the information that you know before each of the presentations. So that's why we're trying just to engage you in with different methods, just to know what you know or don't know, and for you to leave the workshop today with great information as well as some tools that you can implement at your local level. So Hopefully, everybody can participate. We have around 70 participants right now, 73 with the exception of the speakers. So I want to see full participation of all of you. I just see responses from 57. So hopefully we can get all of you participating in the event, and I'll give you a few more seconds. All right. Okay, so let me end the poll here and share results with you. So Lydia is all yours.
Okay, so the question was, which of these benefit from controlling aquatic invasive species? The economy, public health, environment, or all of the above? A really large number of you got this right. There are a lot of benefits from controlling these aquatic invasive plants, and I'll teach you a little bit more about that in my presentation. Okay, I'm going to share my screen. Okay, let's see. My name is Lydia Kennison. I've been with California State Parks Division of Boating and Waterways Aquatic Invasive Plant Control Program as an environmental scientist for about 6 years now. I'm excited for the opportunity to show you all this great program and teach you about some aquatic invasive plants that we're dealing with out in the Delta. So this is an outline of what I'll be presenting to you today. First, we'll learn about the Aquatic Invasive Plant Control Program and some of its key components. And then we've got it split into two teams, one that focuses on floating aquatic vegetation, which I'll refer to as FAV, and one that focuses on submerged aquatic vegetation, which I'll refer to as SAV. I'll discuss what goes on in each of those teams, and then we're going to learn how to identify some of our plants. So to start, I want to explain why invasive plants are important. Caesar touched on it already. This is a flyer created by the California Invasive Plant Control Council. $82 million is a lot of money. Invasive plants are a big issue that take a lot of funds to try to tackle, and that's why workshops like this are so important. The cheapest way to control invasive plants is through prevention, and one of the best ways to do that is with outreach and education. Here's a list of impacts specific to aquatic invasive plants. They can restrict waterways for navigation or recreation, crowd out native plants because they're much hardier and greater resources to grow, and they can also affect restoration sites. Restoration sites are created to provide a healthy habitat for native species, and the introduction of invasive plants can alter their habitat and food web structures for those species. Invasive plants also restrict water flow and can even clog intakes to facilities or agricultural water intakes. Restricting water flow can also cause the perfect breeding grounds for mosquitoes, which is a huge public health concern. So as you saw on the previous slide, impacts of these invasive plants are far-reaching. Our control efforts benefit the environment, public health, and also the economy. In the Delta, we have the Port of Stockton and the Port of West Sacramento who rely on us to keep the waterways open and navigable. If there are a lot of plants covering the deep water channels, ship navigation systems have issues determining what's plant cover and what's land. The Delta also has more than 85 privately owned marinas who rely on boats being able to come in and out of their slips in boat launches. If we find a new invader, we try our best to eradicate the plant, but unfortunately, these plants that you're going to see today are so well established now that we have to kind of move our focus to controlling them. But we do our best with the budget we have every year. So just to discuss briefly the background of our program, we're the lead agency in the California Delta, its tributaries, and Sassoon Marsh for invasive weed control. Harbors and Navigation Code 64 and 64.5 provide information on our jurisdiction and duties. These legislative notes listed here show our start with the Water Hyacinth program and later the addition of the Agarya densa program. And then with AB 763, we became a program that was able to control for multiple target plants. There are a lot of regulatory restrictions and permits that we have to adhere to. Since we do work in a place that has a lot of sensitive species and we operate in waters of the US, we have to take a lot of precautions when conducting our work. For example, the National Pollutant Discharge Elimination System Permit Program, authorized by the Clean Water Act, regulates water pollution. This permit requires us to do stringent tests of water quality parameters, as well as taking water samples to be analyzed by a lab to show levels of herbicide before and after treatment to ensure that we're not harming species or degrading water quality. If you're interested in learning more about any of our regulatory compliance documents, they're all on our website on that resource list that Vivian's already provided to you. So here are some of those sensitive species that I had mentioned earlier. These guys are listed as endangered or threatened, and we do a lot of work to make sure that we avoid impacting them. For fish, we have restrictions on where and when the program starts and ends so that we can best avoid treatments while they're in the area. To do this, we use weekly fish monitoring data from California Department of Fish and Wildlife. And if there's a listed species found in the regularly conducted trawls, we stop our treatments and wait until they're no longer found in that area. If the need for mechanical harvesting arises, we do surveys for giant garter snakes, burrowing owls, nesting birds, and any other sensitive species that may be impacted. We also survey our sites for the presence of elderberry plants, and maps are provided to our field crews indicating where these plants are so that they could avoid them. So this is kind of a cool graph that shows the acres of FAV and SAV treated in a given year. It also incorporates pictures to show what year specific plants were added to our target species list. On the right, you can also see what year we incorporated hydroacoustic mapping, which I'll explain later, and our biocontrol agent, the plant hopper. You'll notice that FAV acres are in green and SAV acres are in blue. Our program is split up this way. We have an FAV team and an SAV team. Treatment methods and monitoring are different for each team. If you think about it, you need very different strategies of treatments for plants that are floating on top of the water compared to those that are fully submerged. Both teams utilize herbicides as their main control method. It's the most affordable and most effective form of treatment that we have right now. Our program places a lot of importance on using an integrated pest control management strategy, though. So we're constantly looking for new tools to utilize to make the program as effective as we can. Whenever we add a new tool or herbicide, we complete in-depth studies and collect a lot of data to prove that we're not degrading or affecting water quality before we operationalize it in our program. So in the middle picture, you can see an herbicide treatment being conducted on water hyacinth. In the upper left picture, you see a drone that's used for herbicide application. And this is a relatively new tool that we've been using. We have contractors that have a great deal of expertise using this technique, and they're able to get into areas that are not accessible by boat and can cover much more area in one treatment. I've been pretty impressed with the drones. They get a really good coverage, and also they're still able to avoid native species like bulrush. On the right, you can see our physical methods of control. Hand removal is up top, but this is very labor intensive and a very slow process. In cases where herbicide treatments can't be used, though, it's an option. And down below that is a picture of one of our mechanical harvesting sites. So while this is in our toolbox as an option, it's very expensive and has the most impact to the environment, so we try to avoid using it. And last but not least, at the bottom left, there's a picture of one of our biocontrol agents. We've worked with the USDA to distribute weevils and then plant hoppers to try to help control hyacinth populations. Unfortunately, even though weevil populations were able to spread, they didn't do as much damage to the plant as we're hoping to see. And plant hoppers didn't give us that great of results either because they didn't really survive that well through the winter. So one of our most important parts of our program is the monitoring that we do. It helps us show our progress and success, as well as let us see where we're not doing that great of a job so we can figure out why. The floating aquatic vegetation team conducts lots of visual surveys. The three pictures at the top are an example of one of our photo point monitoring sites. We visit the same location in the same season multiple times a year and replicate the same picture. This allows us to keep an eye on our sites that we would not necessarily see every day and monitor how impacted that site is in different seasons and in different years. We also collect data of species present in each picture so that we could better understand species at different times. On this slide is also a picture of NASA's Sentinel-2 satellite. So we collaborated with NASA to use the satellite imagery to map FAV cover throughout the whole delta. And this was a big breakthrough because we previously lacked the ability to monitor so much area at one time. NASA created a dashboard for us that lets us visualize current and historical total FAV acres, treatment data, and changes in coverage in any given site. We're still learning to use this tool to its full potential and plan to do a lot of ground truthing this year. Treating submerged plants is a whole different beast than treating floating plants. As you can imagine, things get a lot more complicated when you're treating something that's completely submerged in the water. Physical controls require permits that we do not have yet. Some that we're planning to explore in the future are bubble curtains and benthic mats. We'll have to go through extensive studies in acquiring the right permits to be able to test these methods to see if they have a place in our program. And right now we rely on herbicide applications, and our focus is to find the least risk and most effective herbicides. The SAV team has done a lot of work developing application strategies that show the most efficacy. Our program places a huge importance on adaptive management and is constantly looking for more solutions as we face new challenges. SAV has developed a very sophisticated monitoring technique. So through the use of a program called BioBase, we're able to map biovolume under the water's surface. In order to do this, we use our low-rance units and do a slow drive and transects over our treatment sites. In the upper middle picture, you can see the biovolume map that we get from BioBase. Blue indicates no plants in the water column, as red indicates plants occupying the entire water column. The SAV team then takes that data and creates a change detection map that you'll see on the left. This map shows the change in biovolume and percent cover before and after a treatment. We also couple these maps with rake pulls. We throw a rake from our boat to note abundance and species composition for that point. The SAV team then takes that data and creates those maps that you see on the right. This allows you to visualize how full the rake was and what species were present at each rake pull point pre and post treatment. This monitoring method has allowed us to quantify our success and gives us great insight on planning future treatments. All of these maps are available and included in our annual report, which is on our website as well. So just a summary for the program. We are the lead agency for invasive plant control in the Delta, and we have been since 1982. We utilize integrated pest management and adaptive management, and we're always trying to increase our monitoring tools. So now I'm going to show you a couple of videos that are going to show our program's plants. Afterwards, I'll go through a review of these plants, a really quickly review. It's going to be a lot of information, but all of that information is on our website, so I'll kind of skip through some of it. Let's start with this.
Every year, the Department of Park and Rec maintains control of several different species of invasive plant life. Some of these plants live on the water's surface. We call them floating aquatic plants. These plants can actually look quite beautiful, which is why some people want to include them in their aquatic gardens. Unfortunately, many of these plants have made their way into the Delta and have become invasive. The first plant we'll discuss is called alligator weed. Alligator weed can grow in different environments, so it may appear slightly different depending on its location. It can be found both in the water and on land. The physical characteristics of the plant can vary too. For example, if it's growing on land, it won't have the hollow stem that is typically associated with alligator weed. One of the easiest ways to identify alligator weed is to look for its flower. It resembles a small white cluster, similar to clovers found in lawns. However, depending on the time of observation, the plant may not have any flowers, which makes identification a bit more challenging. Be careful not to let any fragments escape while handling it. as this plant can easily propagate from small pieces. Another feature to consider is the leaves. Alligator weed is often mistaken for water primrose because they both have smooth leaf margins. However, alligator weed has an opposite leaf arrangement, meaning two leaves grow from the same point on the stem, but in opposite directions. This map depicts the distribution of alligator weed in the delta from a previous year. Unfortunately, there will be more locations to add to this map every year. Uruguay water primrose. Identifying Uruguay water primrose can be challenging because its appearance varies depending on its growth stage. When it floats at the water surface, the leaves are round, smooth, shiny, and form a rosette shape. However, as the plant grows taller and emerges from the water, the leaf shape changes. The color of the stems can also change from green to reddish-brown and woody due to environmental factors and age. The easiest ways to identify Uruguay water primrose is by its bright yellow flower, which typically has 5 to 6 petals. Another distinguishing feature is its alternate leaf pattern, unlike alligator weed's opposite pattern. Let's talk about the infamous water hyacinth with its beautiful purple flowers. Water hyacinth have some variation in appearance due to its growth stage and available resources. When it's younger and shorter, you'll notice a bulb shape at the base of the petiole or stem. This bulb contains spongy material that helps the plant float. As the hyacinth grows taller, the bulb elongates and tapers, making it less noticeable. Like the other plants we've discussed, the easiest way to identify it is through its flowers. These flowers can be lavender to white in color and have a bright yellow eye spot. Another feature of water hyacinth is its dark purple to black feathery roots. Sometimes, especially in its early stages, these plants can be confused with sponge plants. The next plant in our floating aquatic vegetation program, the sponge plant. Sponge plant can start off very tiny, and given the opportunity, can grow tall enough to be confused with water hyacinth. The spongy material on the underside of the leaf is even present in the plant's small form. As the sponge plant grows taller, there's less spongy material on the leaf's underside. This can make it harder to differentiate between sponge plant and water hyacinth. Thank you for taking the time to learn about this important issue. We encourage all residents to stay informed about invasive floating plants, report any sightings to our department, and participate in local conservation efforts. By working together, we can ensure that our parks and waterways remain vibrant, healthy, and enjoyable for generations to come.
All right, let's check out our submersed plants now.
Our waterways are teeming with life, but the delicate balance of our aquatic ecosystems is threatened by invasive submersed plant species. Today, we'll identify these plants and explore the impact of these invaders in our delta. Egeria densa, commonly known as Brazilian waterweed, is an aquatic plant species recognized for its submersed, robust whorls, native to South America. Egeria densa has been classified as an invasive species. Its adaptability and rapid growth can lead to the formation of dense mats, potentially displacing native vegetation and affecting local ecosystems. The plant features long, slender stems adorned with whorls of bright green, lance-shaped leaves of 4 to 6 with small white flowers. The next plant is called coontail. This plant is not rooted in the ground. They have heavy stems that temporarily anchor them to the bottom until the current is strong enough to move it to another location. This gives the plant a rough, scratchy texture and a similar appearance to a raccoon's tail. Unlike many submersed plants, this one does not collapse on itself when you take it out of the water. The next submersed plant that we're going to talk about is Carolina fanwort. These leaves are very smooth and soft to the touch. Fanwort is a plant that collapses on itself when you take it out of the water. Because of that, it's sometimes helpful to hold it underwater so that the leaves fan out and allow you to see the true shape. Fanwort characteristics are flat, smooth, fan-shaped leaves which can be red to green and grow in an opposite pattern. The next plant is called Eurasian water milfoil. This plant has a leaf that looks very much like a feather, with 12 or more leaflet pairs on each leaf. There are typically 4 leaves connecting to the same point on this stem, creating a whorl growth pattern which you can see clearly. The plant has delicate leaves that collapse onto itself when removed from water. The final plant we'll discuss is curly-leaf pondweed. Curly-leaf pondweed, Potamogeton crispus, is an aquatic plant known for its distinctive curly and serrated leaves, giving it a unique appearance underwater. This submerged perennial plant is commonly found in freshwater environments such as ponds, lakes, and slow-moving streams. The leaves are arranged alternately along the stem and have a crinkled or wavy texture, providing an interesting visual contrast in aquatic ecosystems. Curly-leaf pondweed is an invasive species in some regions, often outcompeting native aquatic vegetation. Its ability to form dense mats on the water's surface water quality and hinder the growth of other aquatic plants. We're beginning to see a lot of this plant in the Delta, and it's important for us to note and observe these new species as we see them. Together, we can protect our aquatic ecosystems and ensure a vibrant and sustainable environment for generations to come. Join us in the effort to keep our parks and recreational areas flourishing. Respect nature, protect our waters. It starts with you.
Okay, so now we're gonna do a little bit of a review. I'm gonna go over just a key couple characteristics for each one of our plants with you. Most of this you heard in the video, but repetition is always a good way to help us remember. This is a list of the plants that we control for. You didn't get a chance to see the last one for SAV, ribbonweed, because it was recently added to our program, but I've got a couple of slides to show you here. And again, all of this identification information is on our website. So I know it's a lot of information at once, so I'm just gonna stick to like the most key characteristics of each plant. Water hyacinth, purple flowers, large kind of waxy broad leaves. You can see a close-up of the flower here, and you can really notice that central yellow eye spot. Here's just a picture of that spongy material. It's got kind of this bulbous petiole stem thing going on that allows the plant to float at the top, which is that piece right there that you see in this picture. And then just a summary, it reproduces by stolons. I'll say stolons again. That's kind of like a parent plant that creates a runner that shoots off, creates another identical plant, which they can break off and then colonize somewhere new. It has those very unique dark purple and black feathery roots also. This is sponge plant. A lot of times in the advanced growing stages, like in this picture, it gets taller and loses its spongy material under its leaves. And at that point, it can be kind of easy to confuse with water hyacinth. But it's got those spoon-shaped waxy leaves. This is a close-up of one of its stolons that has a flower. It's just a wispy kind of white flower to it. This is a picture of the spongy underside of the leaf. So if you were to actually squish this, you would feel that spongy material. And another picture of the stolon and showing that it's got white roots instead of those purple dark roots that the hyacinth has. This one reproduces via stolons and by seeds. Remember, it's got those light colored roots, and it sometimes can be easy to confuse with water hyacinth. This is Uruguay water primrose. Most of you have probably seen this. It's everywhere in the Delta. It tends to hug the Tule Islands and are all along the bank, but it forms dense mats, and you can see it has a bright yellow flower. So this is really going to be what gives it away. When it's not flowering though, that's when you'll have to kind of look at the leaf arrangement. It has an alternate leaf arrangement, and it's got kind of like a fuzzy stem to it. This is it in its young growth stage. So it's confusing because it can look different at different growth stages. This is kind of like a rosette pattern of very waxy leaves. And you'll see these branches just sprawl out across the surface of the water. This is when it gets to be a little bit more in advanced growth stage. The leaves become longer, tapered, and it loses that waxy appearance. So this one reproduces by fragmentation and by seed. It's got the yellow showy flower, and it can be a hard one to identify because it does hybridize with other Ludwigia species. So that's going to make it more difficult too. And you saw a little bit of alligator weed earlier. This is a very dense mat of it. It's aquatic to semi-terrestrial, so it can go up on land too. And these dense mats, like Cesar also talked about, can create perfect mosquito habitat and slow down water flow. This is a picture of alligator weed's opposite leaf arrangement. So at the same point in the stem, going opposite directions, the leaves are growing, whereas like primrose is alternate. So there won't be a leaf on the opposite side on the stem. This is a picture of a good characteristic to identify the hollow stem. This and then also the opposite leaf patterns that you see in these other pictures. It's got a white kind of pearly clustered flower. It's actually a cluster of a lot of little flowers. And alligator weed reproduces via fragmentation. It's got those pearly white little kind of clover-like flowers. And always remember the hollow stem and opposite leaf pattern. This is getting into our submersed plants. This is the denza. It has whorls of 4 leaves. I'm going to mention whorls again. Whorls is kind of like the number of leaves that grow out from that same point on the stem. So this one has whorls of 4 or more leaves. Here's a close-up of a piece of a healthy plant. It's got a little white flower there. And this is a picture of a rake pole. It just shows how dense this stuff can grow. This is that little white flower I was talking about. So oftentimes, especially on low tide, you'll see little white flowers on the top of the water. And, you know, that's like accompanied by topped out plants underneath the water's surface. So in summary, rows of 4 or more leaves. And this one spreads from budding at the stem nodes. And it's got that white little flower that floats up to the surface. This is curly leaf pondweed. It's a little bit easier to identify because it's got that really distinct wavy leaf. It kind of looks like a lasagna noodle or like a piece of bacon, and it has heavily serrated edges. So if you touch the edge of these leaves, you'll feel it's like pretty rough to the touch. This is a dense mat of it growing. And this is what makes it really difficult to control. It contains these specialized structures called turions, and each plant has a bunch of these, and they drop into the sediment. They can live there for 10 years, and they just wait for, you know, favorable conditions to grow. So we can treat a site, wipe this stuff out, and then, you know, like a few months later, they're just, all of these turions are sprouting. So in summary, they have the alternate and wavy like lasagna noodle leaves. They have very hardy turions, and they spread by fragments and their turions. This one is Eurasian water milfoil. The best way to identify this one is the feather-like leaf. It has whorls of 4 leaves. And so the best, like I said, the best way to identify this one is like if you pull off a single leaf, it really does look like a feather. And this one spreads by fragments and seeds. This is fanwort. So you can see that kind of robust, puffy, billowing kind of shape that it has to it under the water. Here's a little bit more a close-up of that. Here's a close-up of one of the leaves. It's very 2-dimensional, flat, smooth leaf. And this is, it's kind of like pinkish white flower that it has. These aren't seen super often, so that's not probably going to be your key characteristic to look for. So fanwort has opposite fan-like leaves, and it spreads primarily through fragments, but also can spread through seed. This plant is coontail. This is a close-up of it. You can see how serrated and kind of scratchy it looks, and it actually does look like a raccoon's tail. So this is kind of like a bottle brush feel when you kind of rub it between your fingers. This is a rake pole of it, showing how dense this stuff grows. And just to review, it's got that crunchy, heavily serrated feel to it. It looks like a raccoon's tail. and it spreads primarily with turions and fragments, but also by seed. And this is the one that you didn't get to see in the video. This is ribbonweed. It's a new invader. This picture was taken at Delta Marina in Rio Vista. It's really plugged up that narrow entrance to the marina. You can also see this at Hogback boat launch. This is a close-up of the leaves. It's got these very long, flat, ribbon-like leaves to it. You can see the picture on the left shows some of the rhizomes down at the bottom. Here's a picture of the flower. I haven't personally seen the flower, but they are there, and this is one way that it spreads. So ribbonweed has got the long strap-like leaves that can get up to 3 meters in length. It doesn't spread through fragments. Instead, it spreads through its flowers and rhizomes. And most importantly, what can you do? So we're learning how to recognize these aquatic invasive species. Knowledge is key. Always make sure that you clean plant fragments from your watercraft after pulling your boat out of the water. And also avoid purchasing and using plants for decoration that you don't fully know about. A lot of times, if it's a non-native species, it'll have a very comparable native species, and you could use that instead. You can report sales of these invasive plants to CDFA. And again, it is very important to remember and never release these species into the wild. So if you see these invasive species creating a problem, or you're affected by them at your marina or your land, send me an email. It's ais@parks.ca.gov. Please include pictures and exact location. It will help me out a lot to help you. We also have a phone number that you can submit things through. Something that you can do if you're personally impacted, you could do hand removal. You could pull the plants out by the roots and then just make sure that they completely dry above the high water line so they don't make it back into the water. Another thing you can do is hire a diver-assisted hand removal, scuba diver. But it's important to note that only DBW and its partner agencies are legally authorized to use selected herbicides and mechanical and biological control. So here's the website that I've been talking about. Vivian's also provided it to you. It has a lot of good information on it, a lot of FAQs, what you can do, informational sheets on our plants, our annual reports, and our compliance documents. Now I want to introduce my coworker, Michael Rodriguez. He works in another program within our Aquatic Invasive Species Unit, which is the quagga and zebra mussel infestation prevention program.
Thank you, Lydia. And before we do that, I wanted just to mention to all of you that in the chat, I had included access to the all of the resources that you needed to have access to them today, including the agenda as well as the handout. So that's what I have been referring to, and Lydia and other presenters have been mentioning throughout the presentation. Thank you, Michael.
Thank you, Vivian. Thank you, Lydia, for the introduction. So I'll share my presentation. Okay. Okay, so my name is Michael Rodriguez. I'm the environmental scientist with the Quagga and Zebra Mussel Infestation Prevention Grant Program. We call it the QZ Grant Program for short. Our program, let me just move this out of the way. Our program provides funding to California reservoir owners and managers for the purposes of developing prevention plans and implementing activities or implementation activities for infestation prevention. So for the presentation, I'm just gonna go over a few acronyms. So you'll hear me mention QZ Grant Program, that's our grant program. QZ, Quagga and Zebra Mussels, CDFW for California Department of Fish and Wildlife, and of course DBW for Division of Boating and Waterways. And so this is just a kind of a standard presentation we do for overview of our grant program. So here we have our QZ grant team. So our unit chief, Eddie Hard or Edward Hard, and my supervisor, Kara Roderick, the senior environmental scientist, Timothy Giles, my colleague, our park and recreation specialist, myself, the environmental scientist, my colleague, Brandy Vang, our environmental services intern. And it's a small but mighty team. So here's just an overview of what I'll be discussing. I'll go over the funding for our program, who benefits from the program, who is eligible to apply for our grant, what are risk factors that we consider during applications, and some general updates we have regarding our grant program. And then I'll have context and resources at the end. But again, those are also included in that online resources document that Vivian has provided in chat. So here's our funding overview. So our grant program is funded through Harbors and Navigation Code and through statutes and regulations. And so we have the mussel fee sticker, and that's for our state, the state of California. Any boat that is registered with DMV and boating in freshwater is required to have the California mussel fee sticker. And those funds go into this grant program, and 85% of those funds get used for our grant specifically to provide back to reservoir owners and managers so that they can create prevention plans or do implementation projects. And 15% of the funds also get used for CDFW quagga and zebra mussel prevention. And so here's an example of the mussel fee sticker. This is the one that is currently active. It expires at the end of this year, and then the new sticker will be rolling out. It's a 2-year sticker. It follows the same color coding as the California DMV boat registration. So red with red, and then next year it'll be a different color and they should be matching. It is a separate transaction. We did have some confusion with that. In 2021, we made it a separate transaction because we had a requirement from US Coast Guard to not have it tied to vessel registration. And so it's a $16 separate transaction. And it can be purchased either online at DMV field offices or at AAA for any AAA members. And so who benefits from the QZ program? So the funding from our grant program doesn't only help reservoirs to stay mussel free, but it also keeps the cost of implementing prevention projects from being passed on to boaters at the reservoirs, such as offsetting costs for boat inspections. And it can also help maintain tourism. You know, you don't want to have quagga and zebra mussel shells all over your shores, or unfortunately closing a reservoir down due to an infestation. And ensures infrastructure such as water intakes at dams are not encrusted, and ensures ecosystems maintain a healthy balance. Quagga and zebra mussels can be very aggressive competitors for native mussels and native species. So who would be eligible for a QZ grant program? To be eligible for the QZ grant program, the reservoir needs to be mussel free, the reservoir needs to be open to the public, and the applicant needs to be an owner or manager of any aspect of the water in the reservoir. And you would need all 3 of these requirements to be considered as an applicant for our grant program. And so then within our grant program, we have 2 different types of projects. We have tier 1 projects and tier 2 projects. Tier 1 projects, so if you are a reservoir owner or manager and you do not have a prevention plan in place, you can apply for a tier 1 project, and it would be funding to create a prevention plan. So the prevention plan is a CDFW plan that they provide the template for, and you would be applying with them for the prevention plan, and we would provide the funding for that, and we allow up to $200,000 funding for that. Tier 2 projects are the actual implementation. So you have the prevention plan, now you're going to do the actual work. That means inspections, you're going to do decontamination, You may even want to request funding for early detection muscle monitoring. That would be implementation projects, and we allow up to $400,000 funding for that. And here we can see examples of tier 2 projects we have funded in the past. So inspectors or ramp monitors, both human and canine, for inspections and decontamination, mobile and stationary decontamination equipment, mobile decontamination equipment such as like pressure washers, early detection muscle monitoring and water chemistry monitoring, both the personnel, time, and the equipment, decontamination or hydro pads that collect the runoff from spraying the boats during decontaminations, and post or board signage for outreach. So what are risk factors considered when applying for the grant or when we're reviewing applications for the grant? We consider a lot of things. We work with an interagency team, but here you can see kind of a map provided by CFW that shows all the quagga and zebra mussel sightings. And so we consider, of the many things, proximity to infested water bodies, water chemistry, is the reservoir open to the public, or beyond that, how many boaters or events do they host, and how many ramps or access points does the reservoir have? Is there a lot of private access or areas where boaters can kind of get past inspections? Here's a program update from last year. So last year we opened our 2024 grant cycle. We received 10 applications from 9 applicants, and 10 projects were funded, and we awarded about $2 million. The image to the right is a map showing all the reservoirs funded for the 2024 grant cycle. And this is also available on our website in the link that I'll show at the end of the presentation, but should also be included in your online resources. Here's an overall update for our program. So since 2014, we have funded 117 projects, 71 unique reservoirs. So we say unique because we do get a lot of returning grantees and returning reservoirs that apply every other year, and 45 unique agencies with a total amount of about 26.5 million awarded since 2014. And the image to the right is showing a map of all the unique reservoirs funded up through the 2024 grant cycle. The 2024 grant cycle was another competitive year. We received a request of $300,000 over what we had available. And so we're hoping to just keep up the good work and hoping to continue getting good applications. So for our current grant cycle that we just opened, so we did open the application window March 10th. It's closing April 18th, so it's currently open for anybody that would still like to apply. And we will have our interagency review in probably around June-ish, July, and we're expecting the grant year to begin in August 2025, and it'll be a 2-year grant ending in August 2027. And here is that resources page I mentioned. All these links are also provided on the online resources document Vivian provided, but it's our web link to purchase the Mussel Feast sticker, our web link to our website, and our web link to our mussel information page, I believe. And that QR code there is actually straight to purchasing a mussel sticker. And with that, I will end my presentation. Thank you.
Thank you so much, Michael and Lydia. And before we start with the question and answer section for these 2 presentations, wanted just to recap a little bit about what we just talked, right? As you saw at the beginning of the presentation, we have people from many, many backgrounds, not only anglers, boaters, marina, yacht club operators from different water district, the state agencies, public agencies, also potential grantees for the Quag and Zebra Mussel Grant Program. That's why we're providing that information right now, because you would be thinking, well, I'm a boater, what I should care about the grant program. But you should care because we're telling you how the mussel sticker fee is being used, and Michael gave fantastic statistics about how the state is trying just to prevent those species from going into other locations. I'm going to start, Lydia, going back a little bit about a couple of questions that I have for you that I have received from the chat. So, for example, let's say that I'm a boater or waterfront owner or a marina or yacht club operator, and then I'm in the Delta and I see some of the plants that you perfectly described. The question would be, can I call you and can you come to my property or my marina or my facility and treat those immediately? Like I call Lydia and said, hey, Lydia, look at my backyard, our waterway. It's just not in good shape. What are my options?
Yeah, so that website that I listed on the resources page, the dbw.parks.ca.gov, send an email there. I'm actually the one who received those emails, and I'll pass those off to the leads of both the SAV, FAV programs. And we do a great job of contacting you right away, having somebody go out and look at the issue. And then, we usually are able to address complaints right away. We're always working with a different budget every year, and so that may affect it, but absolutely give us an email and we'll do the best we can to help you out. We do prioritize marinas, so do reach out to us.
Thank you. And also I understand our audience already understood that is not we have to go through so many federal, state, and local permits. So it's not like one day we wake up and just go wherever we want. We have to follow also some of our permit requirements and limitations, correct?
Right.
So we have 3 questions in the chat. The first one is from Noriko, and it says, a question to Michael, what is in the mobile decon water tank?
Typically for decontamination, we mostly use hot water. So it's hot pressure washer up to, I believe, 140 degrees. And that's usually used for a lot of local agencies for decontamination.
Thank you. And that question is also going to be addressed by Catherine Mandela later on. Thank you, Michael, in more detail about that kind of infrastructure. Jake mentions, hold on for a sec, because I moved too fast here. Where again can we find the map of infected bodies of water? And Jake, when you talk about infected, are you talking about the presentation that was related to Lydia? In other words, like the Delta ones, or you're talking about the Quag and Zebra mussels? If you can please unmute yourself and specify that. Quag and Zebra, perfect. Thank you, Jake. So we will show you again the map in Catherine's presentation. So you will see that specific information there, and we can also send you the link. Excellent. Now we have Carlos, who's saying, is there any program for checking boats that stay in the water? And I'm assuming, Carlos, are you asking that question in reference to Lydia's portion of the presentation? In other words, the submerged and floating plants, or to Michael? If you can please specify. Mussels. Oh, good. Okay, great. And that one is also going to be covered by Catherine. So hold on to that question too. Then Chris Berry saying, couple of questions. What is U.S. Fish and Wildlife Service posture on oversight of the Department of Fish and Wildlife policies, invasive species and importation, specifically regarding bullfrogs? I don't know, Cesar, if you want to respond to that question.
Yeah, where are you seeing that question? Is it
in the chat? And I can just you can just check the chat. Let's see if it was just directed to me. Yeah, it was directed to me. So it says, what is U.S. Fish and Wildlife Service posture on oversight of the Department of Fish and Wildlife policies on invasive species importation, specifically regarding bullfrogs? That's the first question.
Well, I would have to look more in specifics about, the history in terms of that regulation, but from a collaboration perspective, the Fish and Wildlife Service works closely with the state of California on all invasive species issues.
Mm-hmm.
And specifically relative to bullfrog, I mean, bullfrog is pretty ubiquitous already in a lot of places in California. Like I said, we would coordinate with the state of California. So, for example, if there was a specific moratorium by the state, definitely, obviously, we would agree with that regulation. And I would say that we would work with the states on not just bullfrog, but any sort of regulations that they're considering imposing in terms of stopping the importation of injurious wildlife.
Okay, and the second question is, what are regulators doing about the rampant internet trade in invasive species?
Well, I brought an example there early on in my talk about the Marimo mossballs, and those mossballs were tainted with zebra mussels. So that was one example of where a bunch of agencies, both federal and state, needed to work together in order to not only stop, at the federal level. The Fish and Wildlife Service is working with USDA at our ports of entry to try and stop the initial importation of those mossballs, but then also the federal government working with individual states. So, for example, some of those mossballs were found in stores in Oregon, Washington, and California, and in all those cases, the Fish and Wildlife Service collaborated with those individual state wildlife agencies to collect those specimens and quarantine them and ultimately dispose of them. So, as I stated in my talk from the very beginning, is that handling invasive species kind of takes a village to handle invasive species. It's not just one agency that has unilateral authority to do everything. It's a cooperation and coordination of many agencies in order to get the job done.
Thank you. And this question is for Michael. Are reservoirs the only eligible bodies of water for the Quaggan Zebra grant? This person, Savannah, works at the river park. At a river park, sorry.
Yeah, so I believe that reservoirs are the only applicants right now. So freshwater reservoirs, and you would have to be the owner or manager of the reservoir.
May I add to that really quick? However, those, though the QZ grant is specifically for dammed water bodies within California, there are federal grants, and sometimes the California State Marines, the Fish and Wildlife Commission, will also have grant money for water bodies that aren't associated with dams, like rivers and delta and that type, or private as well, just to throw that out there.
Thank you, Catherine. All right, so we can take a couple of more questions before we go into the break. Actually, we're doing fantastically well in terms of the agenda. This is the time for us to take a break unless anybody has a question started from a bird's eye view perspective about the impacts, and then we specifically move into the delta to talk about the floating and submerged aquatic invasive plants that are part of the control program led by the division. And we also talk about one of the grants that have been created by the division in terms of the quag and zebra mussel infestation program, because some of you will benefit from that grant, but also to tell you how that fee that you're paying, meaning voters are being or is being used to create those grants to control those species and or stop the spread of those two species. So I don't see any more questions. Right now, what I'm going to do is I'm going to start our timer, and I'm going to set up a timer for 10 minutes. So I'll see you all back at 10:45, please. So make sure you can stay connected, leave your cameras off or on, and we'll be back in 10 minutes. We have 3 more minutes to go, so hopefully you guys start coming back to your computers. Right, 1 more minute to start. And Catherine, before we start with you, there is a question in the chat for Lydia, so we'll go over quickly about over that one, and then it will be your turn and Lina's turn, okay? All right, we just have almost 20 seconds for our break. I want to make sure everybody is back and we can continue to fulfill our agenda full of great, great information. And before we turn the presentation over to Catherine Mandela with the Department of Fish and Wildlife and to Lina with the State Lands Commission, we have one more question. Ooh, click. All right, let me just stop the watch here. Great. So we have one more question for you, Lydia, that we just received during the break from Jack Bettencourt, and it says, what effects does the use of herbicides to control water hyacinth have on the overall environment? You're muted.
Sorry. Yeah, so the effects to the overall environment are very minimal. We've done a lot of residue studies that test, how much is going to the water after like a foliar application, which is very little. So that's what's really causing a lot of these things to be in the water are a lot of use by agricultural lands and rural areas that water drains into the delta. Oh, now you're muted, Vivian.
Thank you. Thank you, Lydia. Hold on for a sec. It's somebody else is saying, I'm going to have to leave early. My question is, how will the boat banding program be improved? Oh, that's more like for the next question. So we'll keep that question for the next presentation. It says, my boat has not been in the water for several months, and if I go to a banding site at a lake, I have to stay out of the water for another 30 days. Also, will there be future coordination between lakes, at least those that are on the same watershed? I speak about New Melones and Tulloch. There is much room for improvement. My email is listed on my reservation form today. And that's a question from Robert Baccaro. I don't know, Catherine, if you want to respond to that question, since Robert has to go, even though I understand you're going to cover that later on, but since he has to leave early, can you just quickly mention that?
Could you read the full question for me again, please?
Yeah, and let me copy it here before we finish here. So Robert is saying that his boat, my question is, how will the boat banding program be improved? My boat has not been in the water for several months, and if I go to a banding site at a lake, I have to stay out of the water for another 30 days. Also, go ahead and respond to that question.
Yeah, there's really no simple and fast response to that. Each agency has their own policies, so it's going to depend on the already established program that's already at that water body, where some it's like I said, there's a lot of factors to the reason why there are certain lakes that have closed, certain lakes are having this 30-day banning period, and then there's other lakes that already have a program where you can go through an inspection and decontamination program. The ones that don't have a robust inspection program are requiring this 30-day banding period because they don't have a program in place to be able to thoroughly decontaminate or inspect the boat so that you can launch that day. This is something that's taking time, and I'm going to need more of my presentation to get into the reason why it's going to take more time. But again, best thing to do is to contact that specific water agency. If you call that lake themselves and they say, well, call Catherine, there's not a lot I can respond to other than what I just did. Also, with the fact that they have to have them contact the water agency that has the jurisdiction of that water body. I only cover the Bay Delta region, and I do get a lot of questions coming from outside of that region, so I don't have specific knowledge of lakes programs outside of my region. So I just wanted to bring that up. Hopefully that answered your question. If not, I can provide you, if you want to offer your email, I can provide you a more in-depth response to that.
Yeah, and I already captured Robert's question and his email, and I'll make sure that I email you that question so we can take it from there as well. All right, so excellent. So now we're going to switch into the quagga zebra and the new invader, the golden mussel program. We're going to talk about the biology, how they got here, among other important information. So before we start into the details, excuse me, I'm going to launch another poll. And I want, of course, everybody please to participate. So right now the poll is in your screen. Go ahead and please start reading it and responding, and then we will share with you the response.
Pretty good. Lots of people know stuff already.
And if you don't know, that's totally fine. Remember, every day is a good day to learn something new. That's why we're putting together these workshops. And I'll give you 30 more seconds. So we have so far a participation of around 60% of the participants. So please make sure that you all participate. All right, 10 more seconds, and then we will end the poll. All right, so let's see. Catherine, all yours.
So yes, which freshwater mussels found in California have byssal threads? And for those of you that don't know what a byssal thread is, they're the little hairy things that they'll attach to things with, like rocks or docks, that type of thing. And it looks like a good majority did understand that question. So yes, the answer the correct answer is zebra mussels, quagga mussels, and golden mussels are going to be the only freshwater mussel in California that would have byssal threads.
Okay, excellent. So now you can start sharing your screen and take it from here.
Let me know when you all see it.
Not yet. We'll let you know. Yes, now we see it.
Awesome. So my name is Catherine Mandela, and good morning. It's still a morning. It feels like it's afternoon already, but thank you for all being here. I'm the environmental scientist for the Bay Delta region for the California Department of Fish and Wildlife, and I'm the aquatic invasive species specialist. And though I deal with some other species besides mussels, the ones I will be focusing on today are the golden mussel and then the quagga and zebra mussel, and more detailed. I mean, you've been getting a lot of it already, but like Lydia said, repetition is good. So I do want to start off by explaining CDFW, which is California Department of Fish and Wildlife's role. Basically, we are the lead agency for the mussel infestations and the mussel program, both the quagga and zebra mussel and the golden mussel, when it comes to recreation and creating prevention plans and coordinating with other state agencies. DWR, which is Department of Water Resources, they're the lead agency when it comes to waters associated with the State Water Project. But to get more into what our role is at CDFW, we also offer vessel inspections and decontamination training. We, like what we're doing now, assist agencies and prevention plans for the regional programs. And this is according to our code, Fish and Game Code 2301 and 2302. 2301 has to do with infested waters, such as in Southern California, and 2302 is the non-infested waters. We also coordinate with the California Department of Food and Agriculture, particularly when it comes to quarantine vessels coming into the state. And then we also work with law enforcement, not only just our wildlife officers, but also other law enforcement throughout the state. And these are the prevention efforts that are in California and all the agencies. And you're seeing a lot of us here right now. So besides us, we also have, like I said, Department of Food and Agriculture. They also help sometimes identify organisms that we haven't seen yet. I've taken samples to the Food and Ag, and they'll be able to look up ones that we've never seen in the state yet. California Department of State Parks also helps us, the Division of Boating Waterways. You just saw Michael give his presentation on how one of the ways that they help. Department of Water Resources, they're also out monitoring a lot of water bodies associated with the State Water Project. And just to know, there are some reservoirs that are part of that State Water Project. Some examples are Bethany Reservoir and Del Val Reservoir, both in the South Bay area. And then there's the local and federal agencies. And again, it takes all these agencies to make a program work. So prevention and containment programs are a major role in what all of our agencies are creating to prevent the spread. And I don't need to elaborate on this because you've seen this a lot, and I'm going to get into more detail as we move into this presentation. So these organisms right here, like I said, there's a lot of organisms out there. But what I'm going to focus on are the top three. You see the golden mussel, and then you see the quagga and zebra mussel. I do want to point out these other organisms that you see on the lower part of the screen, because a lot of people confuse them. We actually do have freshwater native mussels. And the two in the green zone there are the two mussels that you'll see in our area, particularly the California winged floater. And you'll see our native mussels are much larger than the quagga and zebra mussel and golden mussel, and they do not have byssal threads. So if you're not familiar with what a byssal thread is, look in the photo in the upper right corner. That's a quagga mussel, and you see the guy's thumb pushing it off to the side, and you see those little hairy things attached to the rock. They're steadfast, and they're really, really durable and really stick. In fact, you can knock shells off, but those byssal threads will still stay there. You will not see that on any native species. So you have the California winged floater, and then you also have the Western pearl shell. And the Western pearl shell, you don't see in most of California as much as you do more in like the northern parts of California and maybe in parts of the Sierra. But if you're in like the Bay Delta region and in the valley, you will see examples of the California winged floater. So if you see them, jump for joy because they are struggling to deal with all these invasives. I do want to mention Asian clam because so many people, we think we've explained, you know, the identity of these mussels really well, but Asian clam are pretty much everywhere. If you go all throughout North America, you'll see them. I was just back east and I saw them in the Potomac and in the Shenandoah and they're in Florida. They're all over and they have those stripes on them, which people will confuse that of the stripes that we tell people to look out for on quagga and zebra mussel. But they do not have byssal threads and they're going to be in the sediment. Now, here is some biology that I want to discuss with these three, Q, Z, G. We've added the G there, acronyms. Hopefully we don't have to add any more acronyms to this, but for now, it's the quagga, zebra mussel, and golden mussel biology. There are definite differences between the golden mussel and the quagga and zebra mussel. They're different species, and they're from different parts of the world. I want to share, it's very technical, but is somebody trying to say something? I'm hearing voices coming through.
No, I'll make sure that everybody's muted.
Okay. So the technical part of it is going to help explain why there are closures in.
Catherine, if you can just unmute yourself, sorry.
Okay. So let me get back to here. Okay, are you hearing me okay?
Yes.
Okay, good. So I'm going to start with the life cycle of quagga and zebra mussels and also golden mussels because they're very similar. Their life cycle begins as a planktonic little larval stage that we call veligers. And when they're about, well, in the case of golden mussels, when they're about 3 weeks old, they'll start developing a shell. For quagga and zebra mussels, they'll be about 4 weeks old before they start developing a shell. And then they can, depending on the nutrients levels and the environmental circumstances, their growth can be exponential or, their growth is exponential anyway. But the warmer and more nice, like the California climate, the faster they're going to grow, which means their sexual maturity rate or the age can come sooner or later, depending on where in the world they are. Generally around 6 months to a year, they can become sexually mature, both with quagga and zebra mussel and golden mussel. Golden mussel probably a little faster. To be frank, there's a lot we still have to learn about golden mussel. There's some details we just don't know yet, but we're in the process of learning that. And I'll get into some more details of that later. But their lifespan both is about anywhere from like 2 to 5 years, but we've even heard and read documents about golden mussels living as long as 10 years. Now, let me get into the chart there on the right side. This is where it's going to be key as to why we have closures in California right now of a lot of reservoirs.
Catherine, sorry for interrupting you. There is a square on top of the area where the origin. Okay, got it. Thank you.
Awesome. So let me start with quagga and zebra mussels. So they can handle calcium levels, which depending on the geology of the area, most of California has enough calcium in in the geomorphology to support quagga and zebra mussel, except for in the higher areas like the Sierras, parts of Northern California. I know there's other states here. I can't speak for those, but they don't really like a lot of salinity. Quagga mussels can handle a little more salinity than zebra mussels. They can handle up about 5 parts per thousand, which if you think in terms of the delta, once you get to about Antioch, after Antioch, it becomes too salty for them to survive. However, I do want to point out that even quagga mussels, I did a study probably about 10 years ago now to see how long it took a quagga mussel to die in salt water. And I had taken samples from like Antioch, then like Martinez, and then like under the Carquinez Bridge, which is about not quite full on salt water. It's about 23 parts per thousand, which if you take the ocean, for example, it's like 32 to 35 parts per thousand salinity. So basically, it took 2 days. There was one mussel that survived 2 days in that saltier water. So for boaters that think, oh, I can just go take my boat out to, if, you come from Lake Powell or somewhere that had quagga and zebra mussels, and you just go boat out in the ocean, there's a couple of issues with that. One, it may not kill them right away. And two, they're a mollusk. So if you think of salt being poured on a snail like people have done, and it froths up, that's what's happening to quagga and zebra mussels. And even it's a little different with the golden mussel, and I'll get to that in a minute. But quagga and zebra mussels, they'll froth up and die, and that will cause it'll gunk up your engine. And, I've heard of boats having to go into dry dock and have a very expensive repair. So that isn't a solution to decontaminating your boat, which I'll get into further later. But quagga and zebra mussels, they can handle a lot. They like to spawn, and their temperatures can range from 34 to 90 degrees to be comfortable. So, in the Great Lakes, which they were first discovered in the mid 80s, they do see die-offs in their harsh winters. But there's always some that still do survive. They spawn when water is warm enough. Unfortunately for California, that's most of California. The water is warm enough for them to spawn. Though they do slow down in the wintertime, they don't completely stop. They don't like, they're going to need some oxygen in the water. So I don't really need to get too much into the depth of that. But in terms of depth, they can handle pretty deep water. Now I have 164 more or less feet that quagga and zebra mussels can survive in, but I do know of people back east in the Great Lakes area saying that they've found live quagga mussels as deep as 260 feet. They mature based on their size. So if the conditions are happy for them and they grow faster, they can sexually mature faster. And their origins are the Caspian Sea area. They did infest Europe around 300 years ago, and then they eventually made it to the U.S. through ballast water and shipping traffic into the Great Lakes, and they've spread westward since then. We first noticed them in California in 2007. Lake Mead became infested with them, and because Southern California gets Colorado River water, they ended up with them, and they were seen relatively quickly after that happened. There's a map to show you this later. Now I want to go to golden mussels. Since the discovery of golden mussels, we first discovered them in October, just this last October. The spooky thing about golden mussels is that they can handle a wider range of everything. They can handle more polluted water. They can handle more saltier water. They can handle less calcium, less oxygen. They can just handle a lot of more unconducive to life environments than other organisms. The only thing they don't like is really deep water. I think from what we have learned so far, they don't really settle much deeper than around 33 feet. And they're from China. There's some other locations in Southeast Asia that they've eventually become established and kind of become part of the ecosystem. But unfortunately, they also spread to, and I remember this back in 2005, I remember hearing about it, but around in the 90s, golden mussel infested South America. So Brazil, Uruguay, and Argentina. We have been in contact with the Brazilian scientists to give us some heads up on what to expect and what they've been dealing with so we can learn from them. One of their biggest messages to us was really pay attention to aquaculture and to recreation, which is things that we've already been on top of with the quag and zebra mussel program. So I'm going to move on. These are their impacts. Ecological, there's ecological, economical, and cultural impacts. You've heard this already from the previous presentations, but I'm going to get a little more in depth with this. I'm going to begin with the ecological one. So with mussels, and this is really the case with both quag and zebra mussel as well as the golden mussel, they'll outcompete native species. You can see that native anodonta underneath all those zebra mussels. That photo is actually from back east. So for you folks, I know there's some Floridians here. There are basically in North America, the native mussels are that bigger, flatter. They're all bivalves, which means they have two shells that close together, but they're larger. And a lot, I know they're listed endangered now back east because of the quag and zebra mussel infestation since the mid 80s. You'll see them, attached to crayfish. I've even seen photos of them attached to dragonfly nymphs in the water, and they're quite small. So they cause a food chain collapse. So basically, they filter feed everything, away to the point where even in the Great Lakes, they've created dead zones because they've filter fed everything away that they don't even have anything to eat. And the problem is when they have their pseudofeces, their poop, when they poop out their when they release their feces, it's so, because of bioaccumulation, so full of toxins. Like I have read papers where their pseudofeces or their environment around them is 100,000 times more toxic than their tissues. I mean, their tissues are more toxic than the environment that they're in because they're accumulating all of this. So if those of you are wondering if we can eat them, I wouldn't, just so you know. I know some people think of pulling out the cioppino sauce, but that's not a good idea. The other problem with this clearing of the water is that they create a plant boom, because basically with less nutrients in the water, it becomes clearer. And then that means the plants can photosynthesize deeper, which is a problem, as you've already seen with all the invasive plant problems that we have in California. And what that ends up happening is that there's a boom in plant growth, and then the plants absorb a lot of the oxygen out of the water. So then there's a dissolved oxygen problem. And then again, nothing can survive. So there's literally like dead zones in parts of the Great Lakes. And since are like already kind of like 40 going on to 50 years post this issue in the Great Lakes, what I've heard from them in those areas is that what's moving into these dead zones now are like invasive species like bass and, other loach and maybe some other species that are not the ones we want to return. So that's an example. Also, they cause harmful algal blooms. And we already have this issue because our temperatures globally are rising. That photo you see over on the cultural side there, and that woman on the beach right there, that's a good example of harmful algal blooms. These golden mussels are likely going to only augment our harmful algal bloom problem. And I do want to just say this because it's not shared enough, but you don't want to be that woman standing near that stuff. You don't want to breathe it. You don't want to touch it. Definitely don't let your dog swim in it. Dogs have died from being exposed to this. Humans have gotten rashes. I've heard, I'm just going to share this because we need to know this. I had a gentleman ask me, I was at a Discovery Bay in the Delta, and a gentleman came up to me asking me if I knew what was in the water or something. He had a doctor asking him to go get samples in the water because a lot of the kids in that area they're experiencing some kind of disease that they can't figure out what it is. And it's a neurotoxin, so it can be hard to detect a lot of times. So just know that. And also don't use rubbing alcohol to wash your hands with it. I've learned that it will break the cell membranes down and actually release more toxins. So use warm water and soap if you do get exposed to it. I just wanted to share that because we need to share that with everybody. Anyway, back to the economical part of it. With quag and zebra mussel and golden mussel, they clog things up. We have not seen the effects of this with our agricultural yet. They cause utility prices to go up. They cause produce to go up. Southern California has been infested with quagga mussels for a while. If you're from Southern California, you've been paying more on your utility bills because of this, because of the extra costs, the extra personnel that is required to deal with it. And it's like Arkansas, they've had the problem with zebra mussels in their agriculture, and they saw an increase in their produce prices. So these are things we want to avoid. Culturally, places are closed. A lot of you're already seeing this. You're wondering, why can't I go boating in this place that I've been going to forever, and I have to deal with this 30-day quarantine period? This is all the problems with regards to this, and all these closures. I will get more in depth as I move on to this rest of this information I'm sharing with you. Here's a map. Somebody asked, where can we see where places are infested? This is just California. I'll show you a location where you can get a national. I think it even includes Canada and maybe Mexico with the USGS, but I'll get to that later. I want to focus on just here with the quagga and zebra mussel. This map, it's on our website, which I'll show you how to link as well. All the red circles are where the quagga mussels are. The lower big red circle, all the quagga infested locations are in Southern California. Some of them are not related to the Colorado River system, and they got spread because of boating, recreation, and trailered boats. But for the most part, I want to point out right here, look at that state. For the most part, considering these infestations and throughout the country, because of the program that we have put in place since 2007, it's around when I got hired, we have prevented the spread. A lot of people think, throw their arms out, it's a lost cause. But I can tell you now, because of boat inspectors, because of detection dogs, we have prevented boats with living quagga mussels from launching in the Delta, in Lake Tahoe, Lake Sonoma, Lake Mendocino, and probably some other locations. So doing the programs that we're doing, we can keep this under control as long as we spread this information. That little circle, the little red circle up there in the north, that's our one and only zebra mussel infested reservoir. It's called San Justo. It's near Hollister. It's been closed since 2008, I think, when they found them. That was kind of a random situation of how that reservoir became infested, but I want to keep moving here because there's lots of information to share. I did add golden mussel locations now on this map. So basically they're in the Delta, and I will have a more in-depth one in a minute, but you'll see where they have spread, like near Bakersfield. And this is all, I'm going to go to this next page. These specific dots right here are where we have detected them. The dots in the middle are like just kind of northeast of Salinas there are, that's O'Neill Forebay. We have not found them yet in San Luis Reservoir, but they are there. And then there's a transfer station at that lower location that adult mussels have, golden mussels have been found, that we're heading out to the coastal range. And, it's really kind of a matter of time before they start seeing them in Southern California. So this is where the absences are. Since we learned this, I do have to say, a lot of public's really frustrated with things, but we have, many agencies have really pulled together, and not just agencies, but the public. And I'll show you how you can be a part of this in a minute as well. But all these locations where people have been going and monitoring water bodies and not finding golden mussels. So a lot of effort has been done already, and I'd be curious to see what other states are doing as well. Here's a map to show you kind of a spooky thing. Here are boat inspections in other states where California Delta, the Delta was the last water that boat had visited, and this is just 2024. And you'll see Lake Berryessa there on the top. When Lake Berryessa saw this, they freaked, and they get a lot of boats coming from the Delta. And when they looked into it more, that count of 831 boats, they realized that wasn't an accurate count. There are many other boats that didn't get on that statistic. They said it was probably more up to like 2,000 plus boats that come from the Delta just in 2024. So I'm going to share this video with you. I took this at Ground Zero. This is the Port of Stockton. I want to, are you all seeing it okay? Do you see it?
Yes.
Okay.
Yeah, we're seeing it.
Now, I want to point out here, if you kind of see, this is a mooring line hanging off the dock at the Port of Stockton. Those are the mussels right there. Because the delta is subject to tide, it's really, you can't just walk the shoreline and see the mussels. You won't find them, which is probably why we haven't detected them. We actually did some eDNA testing of archived samples from the spring of 2023 and found the presence of golden mussels. So they've actually been here longer than we realized. You don't see them just walking along. They're going to be down in deeper depths below the low tide line. And they're going to be under things. They are a mollusk. They don't like light. So where we're finding them right now when we're monitoring is under docks, under boats. A lot of marinas in that area are already, a lot of the boat owners are already seeing mussels on the bottom of their boats. So when you are looking for them, keep in mind, you may not just see them along the shoreline. They're not going to be in the light areas. They're going to be underneath things. They're going to be down deeper. So keep that in mind when you are looking for them. We actually throw out substrates. We'll put these plates of plastic on a cable and we'll put them down in depths and pull them up. And that's an easy way for us to be able to see if they're there or not. There's some other methods that we do as well. But you'll see right here a more up close picture of that rope, that line right there with them. And you'll see sometimes they are a little darker, but there's definitely that gold color. Now, we've mentioned there's so many invasive species out there. We've already talked about all these, but just know when in terms of how you're going to deal with preventing the spread, use the worst ones. Quagga and zebra mussel by far are the most expensive and injurious species out there because not only are they damaging the ecology, they're disrupting our infrastructure and just, our recreation. So, there's so many things. Don't let it loose. Don't let your pets loose. There are places that you can, I can tell you the big chain stores like PetSmart, Petco will not take pets that are no longer wanted. But a lot of times the smaller mom and pop shops do. But there are places to effectively rehouse your pet if you no longer can have it or want it. And I wanted to share this because we all said, well, what can you do? What can just the general public even do? There's something called citizen science, and it's a pretty awesome thing. In fact, if you go on YouTube and Google TED Talks and citizen science, there are some really nice TED Talks regarding citizen science and how they help the scientific community. This is how the public can report golden mussels. So this link right here, which Vivian has, you'll see in the documents that you'll be provided on this this training, this workshop. If you go to the mussel page, and quite frankly, if you can't remember this whole long web link, just if you go to Google and go in the search engine, CDFW golden mussel, it'll take you right to our golden mussel page. And in that golden mussel page, you'll see fact sheets, you'll see maps. There's an interactive map. Those maps that you saw earlier in my presentation came from this link. There's a reporting link in there. So you can actually go out, if you're out and you're boating or somewhere along water and you see mussels, you can report them and it will guide you through. It's a very simple, basic data input that you can put in there, and you'll get an email saying, thank you, we received your report. If you didn't get that, make sure maybe you didn't submit it right, but people, some of those blue dots on that map were from the general public saying, I found them on the bottom of my boat. I found them on, this log I pulled up. So that is a very helpful thing. See that link down, or that EDD Maps app? This is a free app you can download on both Android and iPhones. It's a location that also has photos. It's a nice app to have. I have it on my phone. There's others out there, but this is it's not the most up to date, but it is a helpful tool. It does get looked at by agencies, and it also has photos to help you. If it's not a quagga or a zebra mussel, it could be other things. Something that you're noticing in your area that you've never seen before, you can consult that. Clean, drain, and dry. That's like stop, drop, and roll. Like, we all need to burn this into our memory. So clean, drain, and dry. If you clean, drain, and dry everything, wherever you go, it's going to prevent the spread of many different things, whether it's mussels or snails or fungi. There's a lot of different things we can transport just by traveling around. So these are examples. Your boot, your boats, your dog, your mountain bike, your tivas, your float tube, whatever gets in the water. These things can have little hitchhikers that we don't even see on them. Now, what can we do about it? Besides pulling all this stuff off, I'm going to focus primarily. I have a video that I'm going to share with you right now, and focusing on the most injurious one and the steps to prevent the spread of the most injurious ones, for example, quagga and zebra mussel and golden mussel. That, if you do the measures for that, it's going to take care of most everything. So I'm going to share this video with you now.
In this section, we're going to discuss vessel and gear inspection and decontamination. Assume that wherever you go, whether it's local or abroad, you're likely going to be carrying something back with you on your gear. You will want a scrub brush, a pick to pry out areas such as pebbles and mud in your boot treads, and a thermometer to make sure you have the water hot enough to kill organisms. The smaller the organism, the faster they die. For tougher gear, use hot water at 140 degrees. For sensitive gear, such as scuba equipment and data sondes and other such equipment with sensitive probes, use warm air temperature water. If all you have is a cold hose, then use it. It's better than nothing. Have a gear box. It's an easy way to store gear as well as to submerge it in hot water that can be heated with the portable tea kettle. For things like waders, neoprene, scuba gear, use 100-degree water. As a rule of thumb, for sensitive gear, use water as hot as your hand can handle. This can also be done at home in the bathtub. If you are concerned about melting your gear, contact the manufacturer for verification. This is an example of my field gear decontamination setup. This can apply to field scientists, contractors, as well as the recreationist. The photo on the right is my work truck in which I fashioned a hanging rack system with the use of 2 telescoping boat hooks. This is where I hang waders, nets, lines, and other gear. Because this is work-related, I have designated gear that is only used at one water body. But should I have to use the same gear in a different water body on the same day, it's imperative to make sure to kill anything hitchhiking on the equipment. When at all possible, use hot water. The use of chemicals in some cases, such as medical or specific scientific applications, are required, but for general purposes, hot water will do. Chemicals are harmful to us, our gear, and the environment. Vinegar is also a less harmful and less toxic replacement in some cases. The 12-volt tea kettle is how I heat the water. The portable pressure washer with 3 power sources is how I hose down gear, and the portable steamer is how I decon difficult-to-reach places on gear that can handle such heat, such as felt bottom wader boots, boot treads, and areas of boats that are difficult to reach. All of these items are relatively inexpensive and can be found on Amazon, Walmart, and other locations. The portable pressure washer stores easy in any vehicle, has a long-lasting internal rechargeable battery, but can also be run off a 12-volt or an external battery source, like a Jackery. The pressure washer generally knocks most things off, but for more stubborn areas, use a brush, We were just in a golden mussel infested location, so we followed it up with a vinegar spray that we let sit for 10 minutes and then rinsed later, and also allowed the gear to dry out for several days. Sometimes freezing gear overnight is an option, but this isn't always available for everyone, and it doesn't always kill everything, such as funguses. Clean footwear last, making sure to thoroughly clear the tread and lacing. This is where soaking or steaming comes in handy. For smaller vessels, create a system to clean everything off before you leave that location. In this scenario, I use my truck as a mounting station. I clean the back end first and the middle, making sure to get all the nooks and crannies. You'll be surprised where you can find small organisms. Then I load the boat and clear off the nose part that was touching the ground. Then I clean off the back of the truck, and then I close up and leave. I will make sure the vessel is also dry before I use it anywhere else. This location has a cleaning station next to the launch area. It includes a rack, a hose, and a brush that's attached to a cable. It is encouraged for all recreational boating areas to have a cleaning station. When you think you cleaned the boat well off in the water, it's very difficult to get all the small stuff. In this case, I counted 19 little snails, some of which were the very invasive New Zealand mud snail, on this paddleboard after pulling it out of the lake. After cleaning, find a good place to set the vessel to dry. Use a towel if you can't wait for the sun to dry it. If there are no cleaning stations available, another relatively easy option is to get a set of portable sawhorses. They are multi-use, inexpensive, and portable. For larger vessels, make sure to clear anything off stuck to the boat, trailer, anchor, lines, and anywhere else in the vessel. Remember to pull the plug and dry out even the bilge area prior to launching it at any different water body. For a more in-depth training on inspection and decontamination, contact the Pacific States Marine Fisheries Commission. They offer this training for free. The link will be provided in the follow-up. So right here, this is also an example of a YouTube. I mean, there's so much information on YouTube now. Right after the golden mussel discovery and Lake Berryessa amped up their boat inspection program, they put out and created this YouTube. You can watch it if you want to. This is something that for boaters, you're going to be very familiar with, going from lake to lake. But just know there's lots of information out there. Now, I wanted to share these other web links because they're good information that you can answer a lot of your questions that you already have. This first one right here, the CDFW's conservation/invasives. There's other invasives that we have programs for, like nutria and New Zealand mud snail and a plethora of others. You can go to that link and get a lot of information regarding that species. Now, the middle one, the westernais.org, this is where you can go to sign up for the inspection and decontamination training. They have 3 levels. The first one is just, an introductory level. The second one also includes how to properly decontaminate your boat. And for those of you, I know there are people that have been looking to get certified with this. There's even a level 3. That's a train the trainer that most of you aren't going to be worried about that. But this is a good source to go to get more in depth, because their boats are like fingerprints. There's no one boat that's alike. And, the boat owner is going to know their boat the best, but it's important to know where all to look and where all they can be. Because I'll give you an example. It's really hard to get all the water out of a boat. And the spooky thing with this is, and the reason why we have this 30-day quarantine period is because, boats are, mussels, they can survive some interesting circumstances. For example, they can survive out of water for about, 5 to 45 days, depending on the temperature. Obviously, in the summer when it's hot and dry, after about 5 days, they'll dry up and die. But when it's cool and high precipitation, they've been seen to live as long as 45 days out of water. The exact, there's been no study with the golden mussels yet on this, but we're pretty much
treating them like quagga and zebra mussels. They also can survive in water. For example, there was another study I did where there was quagga mussels in a tank of water that had no food, no nutrients in it. And they lived 3 months in water without eating. So when you take a larger vessel, especially, we know there's always water that stays in little areas that we can't even see or reach. That could mean there's that potential that a mussel could be living, through the winter in southern parts of the United States all winter long without eating. So that's why we have these rigorous inspection programs and why there's closures, because if all of California were to become infested with golden mussels, that would just really pretty much annihilate our infrastructure. Agriculture is one of our main sources of income. It's already going to be impacted by this. There's a lot of reasons why, and it takes time for programs to get set up. So please be patient with these agencies that are that are closed right now. They don't have the means yet to be able to. I mean, you can't just you got your funding for your pressure washer, and I can hire 3 people tomorrow for boat inspections. It doesn't happen like that. And because the calcium levels, pretty much anywhere in Northern California, North America, let me restate that. Pretty much anywhere in North America, golden mussels can survive in. There are very few places that won't be suitable habitat for them. So these locations that in the Sierra, maybe parts of Northern California that didn't have inspection programs set up is because they didn't have the calcium levels to support quagga and zebra mussel. Well, that's not the case now with golden mussel. So and trust me, they're all scurrying right now to figure out to get their programs in place so that they can reopen back to the public. So those of you that have been frustrated with this, I get it. We're living in frustrated times on many accounts, and please share that with everybody. The agencies are doing the best they can to get a program up and running so that we can, get back to somewhat more ease of boating in our reservoirs. So just wanted to throw that out there. And then lastly, there were some questions about how you can find out where places are infested or something's there. Unfortunately, most places have some kind of invasive in their water. There are very few places left, but there's a national one, USGS, this nas.er.usgs. NAS stands for non-native aquatic species. You can go to that link in the search engine. If you put quagga mussel, you can specify what state, if you want just Western states, or if you want all states, you can go there. You can even set up an email alert. There's a place where you can put your email in, and it'll email you when a new species arrives in your area. And if you want just aquatic stuff, or you want just terrestrial stuff, or both, you can specify what you want. And I just wanted to point out to reiterate, we actually still have some states that don't have any mussels in them. Why? It's because these programs got put in place. So I think Oregon, Washington, and Idaho and British Columbia are the examples of not having any quagga or zebra or golden mussel. So the more we do this, it takes a village. I wanted to thank you. Just spread this message, not the mussel. And there are some details with regards to how the golden mussels got into Port of Stockton. A lot of people are trying to figure out how could this have happened? Why did this happen? We have Lena Sabayo to explain this with the State Lands Commission.
Thank you, Catherine. And just for the rest of the public, before Lena takes over, we have, Catherine, a couple of questions in the chat that I'll be reading after Lena's presentation. So all of you We just finished about learning about the quagga, zebra, and golden mussels, biology, impacts, and some best management practices depending on our background we can take to stop the spread. And we also learn what the state is doing at this point, as we learn, especially for the golden mussel. But we're also now going to learn the vector, how the specific, in this case, golden mussel got here. So, Lina, the audience is all yours.
Okay. Thank you, Catherine, and thank you, Vivian, and everyone that is listening. So yeah, I'm gonna talk about this very specific vector, the ballast water as a vector for non-indigenous species. And I'm also gonna touch about the golden mussel introduction and some of the assumptions and the facts about this specific introduction. Again, my name is Lina Ceballos, and I'm a senior environmental scientist with the Marine Invasive Species Program in the State Lands Commission. So, vessels as non-indigenous species, or NIS vectors. So we know that shipping is responsible for up to 79% of established aquatic non-indigenous species in North America. And we also know that prevention through vector management is the most effective way to reduce the risk of transfer of these invasive species. So there are two vectors associated to shipping. It's ballast water and vessel biofouling. And for this specific workshop, I'm gonna focus on the ballast water as a vector. I know that many of you probably already know this, but I'm gonna go through how ballast water can move or transfer invasive species. So we start with a vessel in the source port that is discharging cargo. So as a vessel is discharging cargo, it has to take up water to compensate for the weight and maintain the stability. So as it take up that ballast water, that water comes with whatever is in it, all the microorganisms, zooplankton and phytoplankton associated to it. And then the vessel moves with an empty cargo or some empty tanks and full of ballast water to a destination port. And in the destination port, the vessel starts loading cargo. And as it loads the cargo, it has to discharge that ballast water with everything that is in it. So that's pretty much how vessels move species around the world through ballast water. So how do we manage or regulate ballast water in California? So we have two main ways to manage ballast water: ballast water exchange and ballast water treatment system. So what is ballast water exchange? Ballast water exchange is when the vessel has to exchange the coastal water that the vessel upload in the origin port. That water is supposed to have a high number of organisms, and it exchanges that water with oceanic water that has a low number of organisms. And they do that to reduce the risk of NIS transfer. So that's called ballast water exchange. Coastal water exchange for oceanic water. And then the other management strategy is a treatment system. So vessels have a treatment system installed on board, and these are technologies that are designed to treat the ballast water discharges and reduce the risk of NIS transfer. This treatment there are many different technologies, but it could be ultraviolet or like some chemical injections or filtration in combination with any of these other technologies. But the most important part about these management strategies is that they are meant to reduce the risk, but they are not 100% elimination. So everything that they do is to minimize the risk, but there is no completely elimination of the risk. So in California, we have what we call the discharge performance standards. And I'm not gonna go in detail about this, but what it means is that we have a concentration of organisms allowed in the ballast water discharge. And these standards are divided by class sizes. So for example, organisms greater than 50 microns, that includes most of the zooplankton. The standard for that class size is less than 10 organisms per cubic meter that the ballast water discharge can have. Again, for organisms from 10 to 50 microns, most phytoplankton, the allowed concentration is less than 10 organisms per milliliter. So as you can see, there is, and I mentioned before, this is just risk reduction and not completely elimination. So Vivian, can you launch the poll, please?
Of course I can. Hold on for a sec.
So in this poll, I'm going to ask you what you think about how many commercial ships arrive to California ports per year.
So we'll give you again a minute, which is a great presentation because it really makes us understand how tough it is to control, the some of the invaders to arrive to California. There's a lot of work, and due to the size, it's really complicated. So far we have 72%, 75% participation. So we have 20 more seconds to go. All right, let's see. It's all yours, and let me just share the results.
Okay, yeah, so I'm going to answer that in the next few slides. So the Marine Invasive Species Program in the California State Lands Commission is a multi-agency program, and one of our main objectives is regulate the shipping industry and create and amend regulations based on data and science that we analyze patterns and vessel arrivals and ballast water discharges throughout California, and we use that to amend and create regulations. But the part that I want to focus today is our inspections program. So to answer the poll question, California receives about 10,000 arrivals per year, and that has been a very relatively steady number for the past 12, 15 years. And we, the Marine Invasive Species Program, have a mandate to inspect at least 25% of those arrivals. That is a lot of arrivals to inspect, at least like 2,500. And even though, I mean, it's a lot and it's way more than many of other states that don't have an established program to manage ballast water. So in addition to that, we have like a prioritization process to decide which vessels to inspect. So for the purposes of ballast water, every vessel that is discharging ballast water in California is categorized as high priority for inspection. And we also have inspectors in both South California and North California, in Long Beach and Hercules, that inspect vessels in every single port. So now let's move to the golden mussel introduction and the assumptions and the facts that we know. So one of the assumptions that we think we know is that this was a ballast water introduction. We have heard this in all the previous presentations. And what I want to tell you is that, well, most likely it was ballast water, but given what we know and the biology of these organisms, we think it was ballast water, but could have been through biofouling or aquaculture. The truth is that we don't know. We want to think that we know, but we really don't know. Another assumption is that we know when the introduction occurred. And again, the introduction date is unknown. we heard from Catherine, and we know that the first detection of an adult organism was in October 2024. We have seen that there are multiple age classes in that population. So that means it has been multiple generations there. And Catherine also mentioned that we have seen environmental DNA positives that hit in spring 2023. So if you don't know what environmental DNA is, it's just the detection of the DNA, not the actual organism. So they sample the water and they detect that at some point there was golden mussel there, but we haven't seen the actual organism. And do we know if it was introduced earlier? We really don't know at this point. And I don't know if we're gonna know. Another assumption is that we can determine which vessel was likely responsible. Well, as you can imagine, if we don't know exactly when was the introduction, we probably won't be able to point to a specific vessel. And we're currently analyzing historical data of all the vessels that had source water in all the known established populations of golden mussel all over the world that came to California in the past 20 years. And we're trying to analyze patterns and analyze how much has been the pressure of freshwater, ballast water discharge in our freshwater ports. But this is more like to understand patterns and potential weaknesses, but we really won't be able to point to a specific vessel. The other assumption is that the introduction occurred because of non-compliant discharge. And again, ballast water management reduces the likelihood of successful introduction. However, management does not completely eliminate the risk. And since we won't be able to tell which specific vessel could have brought the golden mussel, we probably will never know if it was due to a non-compliance discharge or just a regular discharge. So, okay, so what are we doing now in State Lands Commission and the Marine Invasive Species Program? So we are in the process of creating these emergency regulations. And what we want is keep reducing the risk. And these emergency regulations, what are gonna require is that vessels that come with ballast water that is low salinity, below 18, and is planning to discharge in any of our California fresh or brackish water ports, now they are required to do, in addition to treat their water, they have to exchange their water. As I mentioned before, they were required just to do one or the other one, mostly treatment. For these freshwater to freshwater discharges, now they're gonna be required to exchange with oceanic water and treat. And why we are doing that? Because adding high salinity acts as a biocide, an additional biocide for these freshwater organisms. And you probably are very familiar with this, but our freshwater ports are considered anything east of Rodeo, so all the Crockett, Benicia, Martinez, all the way to Stockton and Sacramento. Okay, with that, I just wanted to point out to this resource. Our website is in the document that Vivian have shared multiple times, but there you can find these interactive dashboards where you can find all the data about arrivals and the ballast water discharges in California and in all the other Pacific states, Oregon, Washington, Hawaii. So I think with that, I will just take some questions if we have time.
Thank you so much, Lina. And thank you, Catherine. Yes, we do have a few questions, so I'm going to start. with the first question that we received. So let me just go up here. So it was mainly a comment from Robert before he left. He says, it would be good if the Department of Fish and Wildlife could step in and help coordinate the lake inspections between lakes that have banding programs. After all, their funding comes from the fishing and hunting license. So that's like a comment. I don't know, Catherine, if you want just to mention something about that, like why it's not that easy, right?
Well, yeah. Again, I'll just mention that we have been for the last several months. And I was pointing out the fact that it takes time for agencies to be able to hire, to purchase the required equipment to conduct such programs. And it ultimately has to do with the funding that that particular location has, because there's not just water agencies that manage reservoirs. There's also private ones. There's also just smaller local agencies. And it's not always easy for them to be able to just, oh, let's have a program. It takes a while to establish this. And I can tell you now, I know many of them are working diligently on doing that as we speak. So like I said, just be patient with it. We're all working. There's task forces. There's many agencies involved in task forces on this issue.
Thank you, Catherine. Gary Judd mentioned, is there an example of the inspection plates and designs for building inspection points at our marinas?
You know what, that's kind of a good question. And this has been a struggle. Several of us have been working on this. That's not an easy thing to do. It seems like it would be an easy thing to do, but then all the liability things and, issues around liability comes into play. We have been looking into different locations around the Delta that can offer boat decontaminations. Nothing has been, to date, no location has had the bandwidth to be able to do this. There is one location, but you would have to reach out to them. I know it's on the north side. It's in Dixon. It's called Gone Fishing Marine, I believe, that said they would start decontaminating boats. But here's the issue with that. I know there's some other locations too. The Coast Guard has been discussing this to see if some auxiliary locations can be locations to not only decontaminate their own boats, but to be a source for the public to be able to go to for a fee. I've reached out to a couple of boat yards. There was one that used to do it that used to be awesome, but they no longer do this. And that's Eagle Marine used to be a location. They no longer have a decontamination equipment. And I've reached out to some other boat yards and asked, would you be willing to take this on? And basically they said they don't have the bandwidth to be able to take such a huge ask. Like they just don't have enough staff. They barely can get through the clients that they already have. So is this being discussed? Absolutely. I see another question going up there. What was that?
Yeah, let me just go over by order. So Christopher Borjan says, will the CDFW decontamination protocol for quag and zebra mussels now also include the golden mussel? So we talk a little bit that, yeah, they are pretty much the same base management practices, but he wants to know when the specific protocol involving the golden mussels would be available in the website, which you can go ahead and respond to that.
Well, basically, if you go to the golden mussel page, it will have links to basically the protocols for quag and zebra mussel are the same as golden mussel. It's the same approach. It's just a matter of time before the quag and zebra mussel program will incorporate the golden mussel, but basically the protocols are the same.
Yeah.
I hope that answered your question.
Sure. And then this is a question mainly for probably Michael from Robert Janowski. What grants are available for golden mussel prevention?
So at the moment, unfortunately, I don't think we have grants available for golden mussel prevention. The QZ grant is specific to quag and zebra mussels. By statute, it's specifically for dry-sended mussels. That's not to say in the future there may be grants, but at the moment we do not have grants for golden mussels.
Yeah. Remember these mussel got here as we were covering by Catherine and Lena just recently, based on the evidence that we have. It's just October. So the state is moving as fast as we can, just trying to understand all the biology plus the resources that we can start offering. So it may take some time. Carlos Yanis is asking, any program inspection for mussels on in water, hold on for a second, this boats. I don't know, probably Catherine, that's
Wait, say that again. Are there any inspection programs for what, for what?
Hold on. Any program inspecting for mussels for boats on the water?
Yes, there are locations, Lake Berryessa. Well, there's a lot of locations that are still open that are conducting boat inspections. They also have detection dogs. Tahoe is one of them. Berryessa is another. Lake Sonoma is also kind of joining their program along with Lake Berryessa. So there are, yes.
Okay, and Gary wanted to clarify his question. He was asking for designs for detection plates.
Designs for detection plates. I'm not understanding.
Yeah, Gary, if you want to unmute yourself or just type again your question. Oh, go ahead.
The sediment plates.
Yeah, like artificial substrates as we refer them to. You know, quite frankly, well, what we've been using, we've called them quagga condos. I have a photo. Quite frankly, an easy way to do this is to just get ABS pipe and cut them in like 6-inch pieces, drill a hole, get some weight on them so they sink, and put them down. It's a very inexpensive and easy way to do it, and you can, you know, find the materials easy. So does that make sense? They like to be inside and under things and like to be down low. So wherever you hang the plate, make sure it's at least 6 feet to 15 feet down, at least, like off of docks and that type of thing.
Yeah, and Gary's saying thank you. All right, so time right now is 11:59, and what I'm going to do, because I know some of you have other commitments, etc., is I'm going to include the evaluation form in the chat for all of you to please help us to respond. Is one per participant. I know some of you have to leave. We're going to stay here for a few more minutes just to take additional questions. If you need to leave, feel free to leave the room right now. We're going to continue to record this session, which will be shared with all of you later on. But don't forget to fill out the evaluation. In addition to that, I wanted to mention to you that on May 6th, we're going to be offering another similar workshop that is going to focus mainly on salt water. I know some of our partners today are joining us from coastal areas, and they are talking about, hey, can we talk about wakame and all of these other invasive species? So Lena will be joining us to talk about those marine invasive species. And we're also going to bring back again Catherine to talk about the quagga, zebra, and golden mussels in particular, because we want people to continue to be exposed to this message. And Michael as well to talk a little bit about the grant. So that would be the only difference, the component about salt water. So if you need to leave, please leave. But other than that, we will stay here for additional questions. Thank you for your comments, for your attention. We'll send you an email reminder with all the information we discussed for you today. So it's easy for you just to keep it as a reference, and we'll stay here. We got, I think, another question here from Lisa Cusera, and it says, which of the sites is best to utilize for Sierra lakes, and what invasive species are present in those lakes? It may have been mentioned, but I miss it.
Really, both of them are good, both the state, but I also found that if you go to the USGS website, which is the national one, they tend to list a lot more of just a lot more species. Like, I didn't know there were Chinese freshwater jellyfish in some of our coastal streams. Most of the California links aren't going to have those details where the USGS one will, but for the main ones and the bulk of ones and the ones that are most injurious and ones that have programs for, that will be on the CDFW website, as well as the other state, like, Boating and Waterways and DBW as well.
Yeah. We got a question it's more like a clarification from Tracy, and no worries, Tracy, we're here to support you. What was the 15-foot reference testing? I apologize, I didn't catch that.
The 15-foot reference?
Yeah.
I was talking about, I think I was talking about how deep you would want to leave a substrate hanging, the quagga condo, somebody said, because, they don't like light. And especially if you're in an area, like if you're in the Delta and you're influenced by tide, definitely have it as low, not touching the bottom, but as low to the bottom as possible. And having, if you can find, people steal these things. I'm just going to put that out there. Try to disguise, hide it in a place that isn't going to be easy for somebody to pull it up, because people do. I mean, I've gone to substrates and they literally cut the cable off or got a tool to undo the bolts. So, be clever as to where you put them as well. So, yeah. And if you're in a really deep location, have like maybe one at 7 feet and then one at 20 feet, like you can stagger where you have the substrates, the plates.
Okay. Then we got a question for Michael. Hold on for a sec. As an inspector, I have seen many watercraft without their muzzle fee sticker. Are plans placed to get that message to boaters to update with registration easier?
So at the moment, we're still moving forward with having it as a separate transaction. So when boaters renew their registration, they still have to do a separate transaction and get the muzzle fee sticker. And that's mostly due, we had a requirement come down from US Coast Guard. They let us know that we were not allowed to have it coupled together with vessel registration, so we have to have it separate. We've been promoting a lot of outreach and pushing out a lot of messaging for reminding boaters to purchase it separately. But that's the most we can do at the moment, and as well as work with any of our grantees and local agencies on helping reach out to their boaters to purchase the muzzle fee sticker.
Can I add to that too? In our meetings that because that nice QR code that DBW put together, one of the things that we've been trying to address with, when we have our meetings internally with the different water agencies, is to have that at their kiosks and say, hey, where's your sticker? Well, here it is. You can purchase it right now. So if you're in a location and you don't see that information out there, maybe share it with them so that they have that information too.
And we also have flyers for any marinas or water reservoir managers or owners that would like, and it's a flyer that has information on clean, drain, and dry, as well as the QR code on it, and you can keep that at your locations, either laminate it, keep it there for ready to use, or hand them out to people if you'd like to hand them out. You could just reach out to our email. I think that's in the online resources document that Vivian provided.
Yes, correct. And let's see, we have one more question. Okay. I think that's pretty much it. A couple of questions or recommendations for you. Don't forget your evaluation form. We'll send you the follow-up email once the workshop recording is available. I'll be uploading it to our YouTube channel so you have access to it. And then we'll send you more information about the combo saltwater, freshwater, invasive species workshop that we will be offering on May 6th. And we also be sending you the specific contact information for each of the speakers in case you have a follow-up question for each of them. And I think that's pretty much it. I'm going to stop recording here and finish the session for all of you. Let me just do that.